Hello, my name is Michael Malloy. I will be presenting Rift Valley Fever Virus, RVFV. I just want to do a quick presentation layout. What is Rift Valley Fever Virus? I'll talk about phylogeny, characteristics of Rift Valley Fever Virus, the genome type, how does Rift Valley Fever Virus infect cells, its unique immune evasion strategy, viral replication cycle, and the future of Rift Valley Fever Virus at a glance. So to start, a brief history of Rift Valley fever virus. The virus was initially characterized over 90 years ago. The only vaccine approved is for widespread veterinary use and is an attenuated strain that has been associated with significant pathogenic side effects. We talked about the attenuated strain, reducing virulence of the pathogen. So 3. In recent decades, the virus is raising more concerns among Africans due to devastating losses of livestock. Worries of Rift Valley fever virus are increasing due to climate change. Researchers speculate that Rift Valley fever virus may spread out of Africa due to the recent spread into the Arabian Plateau. Researchers have shown renewed interest in Rift Valley fever virus because it's classified as a potential bioterrorist agent. Rift Valley fever virus characteristics. Rift Valley fever virus is a Bunya virus currently endemic to Africa and the Arabian Peninsula. Rift Valley fever virus is known to primarily infect ruminants, though humans can be infected as well. Ruminants include cattle, sheep, antelope, deer, etc. Human infection is less common but is facilitated by mosquitoes as a vector. When large rainfall events occur, mosquito populations dramatically increase, leading to Rift Valley fever outbreaks. So to touch on phylogeny, the species is Rift Valley fever virus. The genus is Phlebivirus. The family it belongs to is Bunyaviridae, and the order is Mononegovirals. The reason I mention this is because I'll be discussing both Rift Valley fever virus and its genus Phlebivirus. On the right, I just included an image that shows us what the virion looks like. We can also see that it is a negative strand RNA virus. We can see the segments on the right as well. Moving on, content of Rift Valley fever virus, or what's inside the envelope. Rift Valley fever virus is a trisegmented, single-stranded RNA genome. It has 3 segments. The segments L, M, and S. We see that segment L and M are negative polarity or negative sense, while segment S is ambisense. Ambisense allows for nucleic acid strands to code for proteins on both sides. And each segment has a cRNA cap on both ends for specific site binding purposes. And when assembled, Rift Valley fever virus particles possess a lipid bilayer enveloped virion with the surface of each particle measuring 90 to 110 nanometers in diameter and being composed of subunits of GN and GC heterodimers, forming an ordered icosahedral shell of 122 capsomeres. So welcome to Rift Valley Fever segment orientation 101. I'm just going to talk about the segments a little bit more. The M segment is a negative strand of RNA. It is responsible for coding the 2 glycoproteins GN and GC. It is responsible for coding the non-structural protein that can be expressed alone as NSM1 or in fusion with glycoprotein GN as NSM2. The L segment is a negative strand RNA as well. It's responsible for encoding the viral RNA. The S segment expresses the nucleoprotein N protein on its negative sense, and on its complementary sense, the positive sense, encodes non-structural protein NSS. The pathogenesis of viral proteins. So viral RNA polymerase is responsible for RNA replication and RNA transcription and is produced on the L segment. GN and GC glycoproteins mediate the virus entry into the cell through its receptors or through the cell receptors. It contributes to the assembly process and likely interacts with the N protein as well. These are formed on the N segment, M segment. NSM1 and NSM2 suppress viral induced apoptosis. These are produced on the M segment. The N protein is produced on the S segment and induces humoral and T cell immunity responses. The NSS is interferon antagonist, which limits interferon-mediated host antiviral responses. How does Rift Valley fever virus infect cells? So the susceptible host for Rift Valley fever virus, it has a wide range of vertebrate hosts, including humans. The tissue tropism involves dendritic cells or mastophages. It looks for the DC sign on a cell surface to bind to. The portals of entry for this virus, we usually see it from mosquitoes. Mosquitoes will puncture through the skin into your circulatory system, giving direct access to these cells. The virion surface displays two membrane-anchored glycoproteins that are arranged in a pentameric and hexameric units to form a unique T12 icosahedral structure. So attachment to the dendritic cell, glycoprotein GN will bind to the DC sign receptor on cell membranes. Glycoprotein GC is responsible for fusion, where Rift Valley fever virus strain MP12 uses dynamic dependent K-vola-mediated endocytosis for cell entry. K-volae are lipid raft domains composed of K-volin. These usually contain a lot of cholesterol and are rich environments. Attachment and replication, moving forward. One of the virion enters the cell, or when the virion enters the cell, the GN glycoprotein begins an acid-activated membrane fusion step. This is regulated by a low pH. Rift Valley fever virus fuses, opening into the cytosol where segments are released. These diagrams describe the positioning of GN and GC. GN is red and GC is blue. These are the glycoproteins found on the virus. Replication cycle continued. Now segments are released. The M segment is negative stranded RNA and makes its way to the Golgi apparatus, producing two effects. NSM is released binding with mitochondria to prevent apoptosis. GN and GC glycoproteins are being manufactured as well. The S segment is a negative stranded RNA. This then translates into nucleoprotein N and undergoes cap snatching. Continuing the replication cycle. Also, the S segment undergoes ambisense transcription to form an NSS protein that is responsible for inhibition of the host PKR. It also infiltrates the nuclear pore and also inhibits initiation of host transcription. The L segment is transcribed into polymerase L. Together, the nucleoprotein and the polymerase L form the genomes and antigenomes of Rift Valley fever virus. Polymerase L is the viral polymerase that is packaged with. So, toward the end of the replication cycle, the negative sense RNA is transcribed into an antigenome, which will not stop due to the prepackaged viral RNA polymerase. The antigenome is used to make negative sense RNA copies. These copies migrate to the Golgi apparatus, where they will meet the GN and GC glycoproteins. The viral proteins are together and the Golgi membrane is utilized by the virus. From here, the enveloped virion will bud from the cell via exocytosis. This is just another image of the Rift Valley fever virus replication cycle. This one pertains more directly to Rift Valley fever virus, though it is also including other virus replication cycles as well. So the unique immune evasion strategy was presented by Ellen Becker in her 2014 study. It's about the nucleoprotein N and how it is very important for replication during many stages of the replication cycle. During replication, N monomers bind along the entire length of the viral genome and antigenome. This encapsulation event protects the viral RNA from degradation and prevents the formation of double-stranded RNA intermediates that could activate the host's innate immune response. The M protein acts as a protection for the viral RNA in the cytoplasm. Ellenbecker wrote that we hypothesize that N binds to RNA in a biphasic manner, and after initial specific binding events, subsequent N monomers bind in a non-specific mode and coat the entire length of the viral genome or antigenome. This is a cloaking mechanism for the mRNA strands. Furthermore, this is her quoting again. Furthermore, we propose that the inhibition of RNA binding to N represents the attractive antiviral therapeutic strategy because several essential steps in Rift Valley fever virus replication cycle involve N binding to viral RNA. Sumarin was proposed because Sumarin has been used in Africa to treat this African disease, although not without some undesirable side effects. I quoted her again saying, "We show that Sumarin in small molecule identified by our laboratory in a high throughput screen as an nRNA binding inhibitor decreases Rift Valley fever virus replication in human cell culture." So continuing our unique immune evasion strategies from Ellen Becker in her 2014 study, the three conserved basic residuals in RNA binding cleft of N were changed to aspartic acid to abolish the RNA binding activity of N. This was their main this was the main goal in this study. The plaque assays were conducted to measure the ability of Sumarin to inhibit production of viable virus over the course of several days. So we can see in the image to the right, the 100 microunits of Sumarin had the best results with the least number of PFUs. So Sumarin decreased the number of PFUs per milliliter by approximately 1 log in 2 and 3 days post-infection and was not cytotoxic to human 293 cells. These data suggest that Sumarin is able to prevent Rift Valley fever replication at multiple stages during virus replication. Sumarin inhibits uh, inhibits our early steps during the viral Sumarin inhibits an early step during viral replication as well as one or more later steps that have yet to be determined. These are my references, and thank you for listening.